Hello, everyone, and welcome to this week's edition of the Extracellular Vesicle Club. This is an event of the International Society for Extracellular Vesicles, and as in several instances in the past, today we have a collaboration with one of our local national organizations. Today it's going to be the Slovenian Network for Extracellular Vesicles. So I would like to introduce Mateja Manček, who is also a member of the board of ISEV and a member of the Science and Meetings Committee.
Thanks, Cam, for this very nice introduction. We are very happy to co-host this EV Club today, and I'm happy that everybody joined and will hear something about our network, but more importantly, also something about scientific work of our member. Let me introduce a very important member of our society, very active member, and this is Mateja Manček Keber. She works at the National Institute of Chemistry in the Department of Synthetic Biology and Immunology. And in her work, she tries to uncover molecular processes in innate immunity against bacterial infections, with focus on human protein MD2, which is a key part in the toll-like receptor 4 signaling pathway. And in the last years, she expanded her research also into the role of MD2 and TLR4 in the sterile innate immunity and oxidative stress. And today she will talk about the role of extracellular vesicles in all of this. Her work was published in 29 research papers. She presented several talks, including at ISEV events, and she was also recognized for her work by the Slovenian Biochemical Society. So I'm very happy to introduce Mateja, and the floor is yours.
Mitka, thank you very much for this very nice introduction. So now I will start to share the presentation. Okay, so thank you for inviting me to share with you our results that we have on extracellular vesicles. And as Mitka already mentioned, the main topic of my work is the research on innate immunity signaling pathways. So you probably all know toll-like receptor signaling. And as you know, these receptors recognize pathogen-associated molecular patterns, which we call pumps. For example, TLR4 MD2 receptor complex recognizes lipopolysaccharide from bacteria. TLR5 recognizes flagellin, and so on. So the binding of the ligands to toll-like receptors induces their dimerization and oligomerization, and then MyD88 protein binds, which forms quite big aggregates, which are called midosomes, and they are necessary for the activation of NF-kappa B transcription factor, and then for the expression of pro-inflammatory cytokines, which drives the inflammation. So, but in the last decade, also toll-like receptors were identified to be important in the so-called sterile inflammation. So that's the inflammation where there is no infection. That means that there must be some endogenous molecules, which we call damage-associated molecular patterns, or DAMPs, which must activate those receptors. For example, one has been already identified, maybe some more also. For example, HGBM1, which is a protein, and it was shown that it can activate TLR4 MD2 receptor. But according to the structure of LPS, which is like this, we believe that the structure of ligands for TLR4 MD2 might be also different. So we started to work with extracellular vesicles now more than 10 years ago, and our first samples of extracellular vesicles we got from Department of Rheumatology, and those were the samples from the patients with rheumatoid arthritis. So we tested those samples and we saw that they stimulated TLR4 MD2 receptor, but it was not shown for the EVs isolated from healthy donors. And these vesicles were not stimulating some other toll-like receptors like TLR1, 2, or TLR5. So then we start wondering what was the difference between the EVs from the patients and the EVs from healthy donors. And one of the processes that drives sterile inflammation is oxidative stress. As you probably know, oxidative stress develops as the imbalance between reactive oxygen and nitrogen species and the antioxidant mechanisms in the cell. As a result, we get oxidative damage, which normally occurs on lipids, proteins, and nucleic acids. Especially unsaturated fatty acids in the phospholipids are highly prone to oxidation. And that is why we decided to induce oxidative stress in the cells by adding calcium ionophore. So addition of calcium ionophore increases calcium concentration in the cell, and as a result, we get higher reactive oxygen concentrations in the cell due to mitochondria. But there are also some enzymes like lipoxygenases and cyclooxygenases which are activated during this process. And after 1 hour of incubation, we isolated the stress EVs from the supernatant using ultracentrifugation. So as you can see from those figures, we have quite heterogeneous population of the EVs because we are not dividing them to small EVs, to large EVs, or to any other kind of specific EVs. So those vesicles were then tested for the stimulation of the TLR4 MD2 receptor. And here we can see that they stimulated NF-kappa B transcription factor, although less prominent as lipopolysaccharide. So if we injected those stress EVs to wild-type mice, we saw increase in IL-6 production in the serum. But if we injected them to TLR4 knockout mice, this elevation of the IL-6 was much lower. Then we performed RNA microarray analysis. And with this, we saw that there are quite some of the genes that are significantly differently expressed from LPS-stimulated cells or control cells. And those genes were shown to be important in the regulation of the response to wounding. They were part of molecular mediators of the immune response, cytokine response, and so on. Then we selected some of them. We selected IL-23 and CCL24, which are the cytokines, and also some proteins that are involved in the immune response, like TNF-AP6 or SOX2. And here you can see that LPS and stress EVs induce IL-6 production similarly in wild type, and that this production was still TLR4 dependent. But IL-23, CCL24, and TNF-AP6 were only increased when the cells were stimulated with stress EVs, but not with LPS, and this production stimulation was also TLR4 dependent. From our previous results, we were thinking that probably oxidized phospholipids, which structurally resemble LPS, might be the ligands for TLR4 MD2 receptor, and that is why we prepared synthetic EVs composed of phospholipids that were identified to be present in the EVs. So we used phospholipids as well as lysophospholipids, and we prepared synthetic EVs of the size 100 nanometers. Later, we oxidized those synthetic EVs with 15 lipoxygenase. As I have already mentioned, lipoxygenases are the enzymes that are activated in the oxidative stress. And what it does, it oxidizes unsaturated fatty acids at C15 carbon when there is a double bond present next to it. So it can either oxidize free fatty acids or esterified fatty acids to phospholipids. So here you can see that when these synthetic EVs were oxidized, there was increase in IL-1 beta expression. Then we decided to oxidize each component of the synthetic EVs separately, and to our surprise, lysophospholipids, which only contain one acyl chain, but not the phospholipids that contain two acyl chains, were activating NF-kappa B transcription factor. And as you can see here, this activation was again TLR4 dependent. So the phospholipids that we use for the preparation of synthetic EVs were from the natural sources. That means that we don't know the exact which acyl chains are esterified to phospholipids. And there's a mixture of all kinds of acyl chains. And that is why we then tested the specific characterized synthetically synthesized arachidonyl lysophosphatidylinositol, which contains arachidonic acid with this C15 carbon. And after oxidizing it with 15-lipoxygenase, we performed mass spectrometry analysis, and these are the products that we identified. So there was addition of hydroxy, hydroperoxy, or either keto group to this arachidonic acid. This oxidized lyso-Pi induced dimerization of the MD2 TLR4 protein complex, similarly like LPS, and it also stimulated NF-kappa B transcription factor. And if this was then incubated with inhibitor of 15-lipoxygenase, the activity was much lower. So this oxidized lyso-Pi also stimulated IL-6 production in TLR4 dependent manner in macrophages. We also detected CCL24 and IL-23, so the gene pattern was similar as with stress EVs. But what was observed in macrophages was that the amount of IL-6 and here of IL-23 was very low. And that is why we then stimulated also the dendritic cells. And here we saw that the amount of secreted IL-6 and IL-23 were much higher than in macrophages. And this implies that dendritic cells might be more important in the response to oxidized phospholipids than macrophages. The amount of lysophospholipids is quite low. So in the synthetic EVs that we prepared, there were less than 5% of lysophospholipids. So there might be a mechanism which could increase the amount of lysophospholipids. What is known is that when the oxidized, when acyl chain in the phospholipid is oxidized, it protrudes to the aqueous phase because it becomes more hydrophilic. So, and this oxidized acyl chain then becomes available to either receptors such as CD36, but these kind of mistakes that are now appear in the membrane can also increase the availability of phospholipids to phospholipases. So what are the phospholipases? These are the enzymes, if we look for phospholipases 2, that hydrolyze phospholipids at SN2 position. And what we get is a lysophospholipid and a free fatty acid. So for phospholipases 2, especially for the secreted phospholipases, it has been shown that they are quite increased in the inflammation. And for example, in the synovial fluid from rheumatoid arthritis patients and gout patients, it has already been shown that activity of phospholipases 2 can be detected. And we repeated those experiments, and also in our samples, we were able to detect secreted phospholipase 2 activity. So then to show that 15-lipoxygenase and phospholipase 2 are needed for the formation of the ligands, we prepared another type of EVs. Those were composed of AAPE. That's because when, as I have already shown, when there's hydrolysis, we get the lysophospholipid, which can be oxidized. So those synthetic EVs were first oxidized, and then we incubated them with some different secreted phospholipases. And you see that in all cases, we were able to detect IL-6 expression, which was TLR4 dependent. Afterwards, we decided also to use synovial fluid as a source of phospholipase activity. So we again oxidized the synthetic EVs, and then we incubated them with the synovial fluid from patients. And you can see that there was increase in TNF-AP6 expression. But to our surprise, we didn't see increase in pro-inflammatory cytokine secretion like IL-6 and IL-1 beta, and we don't really have the explanation why is that difference. So then to further confirm the pathophysiological role of the phospholipases, we decided to use a serum-induced arthritis model. So we took wild type and TLR4 knockout mice, and on the day 0, we injected the serum. And then again, on the day 2, we injected the serum, but we also injected into the left knee human recombinant secreted phospholipase 2A. And then for the 14 days, we were measuring the swelling of the ankles. And here you can see that after injection of the human phospholipase 2A, there was a significant increase in the ankle thickness in wild-type mice, but not in TLR4 knockout mice. And that shows that probably phospholipase can promote the arthritis, because also when there was no injection of the phospholipase, there was no difference in the ankle swelling comparing to wild type and TLR4 knockout mice. So then to conclude what we showed that oxidative stress induces the release of stress EVs. This we also saw because if we incubated the cells with antioxidants such as N-acetylcysteine, the amount of released stress EVs was much lower when no antioxidants were present. Then we showed that stress EVs activate TLR4 MD2 receptor, and that the cells of the innate immune system can differently interpret pathogen-associated molecular patterns and dumps such as stress EVs after TLR4 MD2 stimulation. So we saw this by cytokine expression IL-23 and CCL24, but interestingly, we also saw that stress EVs don't induce tolerance in macrophages. So induction of tolerance with LPS is something that is very known, the mechanism, and it happens that if you first stimulate the cells with the low dose of LPS, and after several hours, you again stimulate the macrophages with higher dose of LPS, the cells will produce less cytokines. And it seems that this is a protective mechanism when we have infection. So, and with dumps and stress EVs, we just don't see this effect. So that's maybe why in chronic inflammation, we see this continuous inflammation. Then we also showed that oxidized lysophospholipids are the ligands for TLR4 MD2 receptor, and that the enzymes like lipoxygenases and phospholipases are involved in this oxidized lyso formation. So at the end, I would like to thank the people that worked on the project, especially my previous PhD student, Tai, and Dushko, who performed mice experiments, and these are our other collaborators from several institutes. And thank you for listening.
Well, thank you very much, Mateja, for that presentation and for this exciting work. I think it's very important to understand how this chronic inflammation is happening, and I really appreciate your contributions here. Given that these stress EVs are leading to inflammatory responses, what do you think can be done to stop this or to somehow modulate the process?
Well, I think here is really important what we see, that there are the enzymes that are involved. So we can inhibit the enzymes like lipoxygenase and phospholipases, and if we do this, we will not prevent the response of our body to infection, so which is probably very important in these cases. So we have a possibility to limit only the chronic inflammation. So, but it's known that quite some inhibitors of phospholipases were already tested in clinical trials, and also of lipoxygenases, but there were quite some of side effects. So there is now only inhibitor of 5 lipoxygenase that is used for the treatment of asthma, but all the others are not in the use. So maybe the combination of both maybe could, lower down the side effects, because if you only inhibit one, then you have the products that are there, and do the harm probably. So I don't know, but that's
achieving that balance, I think, is the goal. So yeah. Well, we do have a few questions that are coming in the chat box. Let's start out with Lucia Languino. Lucia, welcome.
Hi, Ken. Hi, Mateja. Hi, Metka. Thank you very much for the nice presentation and also for telling us about the society in Slovenia. So I'm going to start with a scientific question and then a comment for the Slovenian society. So Mateja, the stress granules, there is actually the first author of the paper that's published quite a bit is from Albania, but she's now in the United States. And it's an interesting question that we're actually trying to address ourselves. So is there known mechanism in your knowledge that correlates the two stress granules and the stress EVs? You know, stress granules are not any typical EV, but I'm wondering if it's a response that the cells do need to have that is similar.
Sorry, I really...
Okay. No, me too. I'm very curious, and there is nothing in the literature. I was hoping you will have some insight.
Yeah, no, the granules, I know that the enzymes like secreted phospholipases are in the granules, and they are secreted when there's inflammation and when the cells are stimulated. But then what is left, and if this, what's left from these vesicles, if this can then have immune response, I really don't know.
Thank you. So I know there is nothing known. So it's a good area of research to start. Okay, and then congratulations to Slovenia. Today I was talking to a person in Austria who was very interested, and I know there is a society in Austria, but it's not linked to your society at all, right? That they are two separated ones?
Yeah, so we are now we are meeting in kind of like all known societies in the European regions are slowly meeting, and we're collaborating on some mobility networks. So yeah, I'm aware that there is one, and I think that we should definitely organize something together at one point, a common workshop or something. Yes, thank you.
Thank you, thank you, thank you. Thank you, Ken, for organizing this.
Thank you, Lucia, for the questions. I have let's see here, a technical question from Connor, who asks, how long were the cells treated with the calcium ionophore before collecting the stress EVs?
1 hour.
1 hour treatment.
Yeah, not more than 1 hour. So if you treat them for too long, then there's no activity of the stress EVs anymore. So.
Okay, so you actually see that goes away.
Yeah, it's really time dependent, and you really need to have more things done and ready so that at the end you get what you want. Yeah.
Oh, that's very interesting. Yeah. Good. And then Metka, I see that you have a couple of questions in here. Do you want to go ahead and ask them?
Yeah, sure. So I was wondering, did you, in these experiments where you've injected serum into the mouse, probably into the joints of some mouse models, did you try to also just inject stress EVs or EVs isolated from disease synovial fluid?
Yeah, we only injected the EVs when we wanted to see if there is any immune response to the EVs. Mm-hmm. And in that case, we saw that there was an increase in IL-6 in the serum. We also done some experiments 10 years ago when we also injected the EVs to promote arthritis. We saw that there was more swelling, but then when everything was analyzed, it was not really significant. But it's true that in these last 10 years, we learned so much about EVs that maybe now, if I would repeat that experiment, much of the things would be done differently. So
always, right? I think this is what you always learn. When you publish your paper, you would just redo it in a better way. So I actually had another quick question. Were you able to characterize lipids on EVs that would be isolated from this arthritis synovial fluid? So rheumatoid arthritis synovial.
Yeah, the problem with these oxidized lysophospholipids is that they're quite unstable and their amount is really low. So in last few years, 3-4 years, when we were working on that, and also a group of Gross was working with that, they are very good in mass spectrometry, and they really pushed the field forward in identification of these oxidized lysophospholipids using mass spectrometry. So, and they were able to detect oxidized lysophospholipids in human myocardia, in hepatic tissue, and so on. But they did experiments on different phospholipases and on cyclooxygenase. So it's a little bit different, but they were able to detect those. And then we also tried to detect them in synovial fluid. We were able to detect arachidonyl lysophospholipids, so unoxidized. And we were also able to detect some of the oxidized lysophospholipids, but they were a little bit different from our standards. So we can't completely say, but they're molecules that are very similar to what we think are the ligands.
Thanks for the questions. We have a question now from Dr. Figueroa Hall.
Hi, I was just wondering, what platform do you use to measure the cytokines from your EVs?
Normal ELISA, nothing special. But we are not measuring the cytokines in the EVs, if you are asking this. We are measuring the cytokines from the cells that were stimulated with EVs.
Okay, I see. Thank you.
Thank you. And a question from Wellington Pham.
So when you adaptively transfer the EV in vivo, currently, what is the best route of injection?
The best route of injection. Well, the experiments that we performed, we injected them intravenously. So in this way, we were able to see the increase in I6, but it probably depends what you want to see and what you want to do. So
because I think that it must be regarded about the stability of the EVs. You know, like in dendritic cells, there are many demonstrations showing that IV injection was not, you know, very optimal compared to sub-Q. So I guess EVs also have some kind of unique way of distribution. So just want to know.
Yeah, I think that if you inject the EVs intravenously, then the majority goes to liver. So what normally happens, that is why I said it depends on what you want. So we already also injected them once into the knees. As I already mentioned, in that case, we saw the swelling that was happening in the knees, depending on what you want to see.
Okay.
Good. Yeah. Thanks for the question. It sounds like then there might be some opportunities to understand better what happens if there is a subcutaneous injection or you know, injection into different tissues of these stress EVs. So a lot of interesting routes of exploration for us. Very good. So do we have any other questions from the audience for Mateja today? All right. Well, I'd like to thank everybody for joining then today and for all of the good questions. Thank you, Metka, for presenting on the Slovenian network, and we want to wish everybody all the best in the network and as the network grows and as the research grows. And most of all, Mateja, thank you so much for sharing your work with us. And we wish for the best as you continue this work and continue to add to the exciting findings from the Slovenian network. So thank you, everybody. Hope you have a good rest of the day and a good rest of the week. And we will see you again soon at an EV Club coming up. Take care now.
Bye.
Bye-bye.